Bob Horvitz, what an absolute pleasure to welcome you to the Bangalore Tech Summit. And it is a great honor to have your presence at this Fireside Chat. So let me start with your incredible journey. You started off as an undergrad at MIT in math and economics. And then you decided to sign up for biology in grad school at Harvard. And you said, don't be afraid to do something new. Tell us about your academic journey.
Well, first, thank you very much for this opportunity, and I'm only sorry I can't be there in person. I would love to be able to do so. To respond to your question, becoming a biologist was something I have to say I didn't anticipate at all at a younger age. In high school, I worked on the school newspaper, and I thought about majoring in English and maybe a career as a journalist. My high school English teacher, in fact, warned me my life would be ruined if I went to a technical school like MIT. I nonetheless went. I don't think my life was ruined. As you noted, as an undergraduate at MIT, I received two degrees, one in mathematics and the other in economics. I wrote my undergraduate thesis in the field of mathematical economics with Bob Solow, who went on to win a Nobel Prize in economics, and I must say, not because of my undergraduate thesis. While I was at MIT, I worked all 4 summers at IBM, and I expected that after graduation, I would continue working for IBM. Computers looked like they had some promise in the 1960s. I think IBM also expected I was going to go work for them. In addition, I was very involved in student government. And when I wasn't thinking that IBM was my future, I was really thinking about law school and a career as a lawyer and maybe even entering into politics. All of that changed when I entered my final year at MIT. A roommate who was a biology major persuaded me to sign up for the introductory course in biology. What he told me was, quote, biology has gotten interesting. Um, I signed up and I liked it. And 6 weeks into my last year at MIT, I went to the professor teaching my biology course. And I said, Professor Leventhal, I'm a senior majoring in math and economics, and I like your biology course, and I'm thinking about going to graduate school in biology. Would I be crazy? And Professor Leventhal looked at me and he said, my undergraduate degree is in physics, my PhD is in physics, and I'm teaching your biology course. You would be starting early. I think that was good advice, and it led me to a view I've since held very strongly. Don't be afraid to change and do something new.
So, Bob, obviously, that's also about the fact that life offers you many, many choices along the way, and it's up to you to make those choices. So that was a great, great way of learning about your journey and the choices you made along the way. So let me now, ask you about your seminal research in developmental biology in C. elegans, which is described as a tiny worm that has a genome with functional counterparts in humans. And this led to the understanding of apoptosis, a process in which cells kill themselves. This research led you to Stockholm in 2002 to receive the Nobel Prize in Medicine along with Sidney Brenner and John Sulston. Do tell us about your discoveries concerning apoptosis and perhaps how these discoveries relate to cancer, because cancer is a huge area of interest these days.
Well, my laboratory has long studied C. elegans. I find myself wanting to say laboratory as I'm speaking with you because I come back to some of my British history. My laboratory has long studied C. elegans. C. elegans is tiny. It's only 1 millimeter long, and it contains fewer than 1,000 cells. A very simple little animal. And Sidney Brenner had introduced C. elegans to modern biology and worked out basic methods for its study. And then John Sulston, with a little help from his friends, myself included, determined the complete pattern of cell divisions and cell fates, the so-called cell lineage, responsible for C. elegans development. I should note that John later helped pioneer the Human Genome Project. And it was our analysis of the C. elegans cell lineage that led to the discoveries in my lab of genes and genetic pathways involved in apoptosis, a process, as you note, is key in human cancer. And in brief, what John and I did was we examined how C. elegans develops from the single-celled fertilized egg. We all start from one cell to the adult, which contains 959 cells. And what we discovered is that not all of the cells that are generated during worm development are found in the adult. There are 131 cells that are generated but are not in the adult. And the reason they're not in the adult is they die during development. This process of naturally occurring cell death is referred to as programmed cell death, also known as apoptosis. And it turns out apoptosis is universal among animals. For example, as a human develops in its mother's uterus, the human embryo forms webbing between its fingers and toes. And this webbing is cellular, and the webbing is removed before birth because the cells that make up the web regions undergo apoptosis and die. My lab identified and characterized the genes and proteins involved in apoptotic cell death. And it turned out that what we found in a worm applied to other animals. The genes and proteins were evolutionarily conserved, and the pathway we discovered was universal among animals and really helped provide a blueprint for understanding the apoptotic process in humans. You mentioned cancer, and importantly, when apoptosis goes wrong in humans, disease results. In fact, I might say that in general, disease is biology gone wrong, and apoptotic disease is no exception to this. Cancer is one of many disorders that can result from dysregulated apoptotic death. For example, too much apoptosis causes cells that should live to instead die. And this is what occurs in heart attacks. Heart cells die. Certain neurodegenerative disorders, brain cells die. Cerebral stroke, congestive heart failure, acute liver injury, aplastic anemia. All of these are disorders in which cells that should live instead die and do so by undergoing apoptotic cell death. There's the opposite side, disorders in which there's too little apoptotic death. And in such disorders, what happens is cells that should die instead live. And too little apoptotic cell death is the cause of certain autoimmune disorders. Cells in the immune system that should die instead survive, and they can then attack living cells in our own bodies. Too little apoptosis also is a major driver of cancer. Most people think about cancer as resulting from too much cell division. Cells just divide and divide and divide, producing a tumor. And that's true, but it's only half the story. The number of cells in our tissues is defined by a dynamic equilibrium involving two opposing processes. First, cell addition by a process known as mitosis, and secondly, cell depletion driven by cell death, i.e. apoptosis. Just as too much cell division can drive cancerous growth, so can too little cell death. It's like a bathtub. If you fill the bathtub halfway up, you can make it overflow by opening the spigot and adding water. But you could also make it overflow by decreasing the rate at which the drain allows the water to drain out. Exactly the same situation with cancer. Cancer is caused by too much cell division and too little cell death, and some human cancers, like follicular lymphoma, are primarily disorders of too little apoptosis, and all human cancers involve too little apoptosis. And it's for that reason that our discoveries concerning apoptosis and C. elegans help define a major fundamental molecular genetic pathway involved in human cancer.
Thanks. That was a great explanation. But maybe I'd like to continue along that path because your Nobel Prize also recognized your contributions to the understanding of another aspect of cancer, the RAS pathway. Tell us about these discoveries.
Yeah, well, the RAS pathway came about by our studies of cell signaling during C. elegans development. Cells often communicate with each other, for example, to control the process of cell division. And we discovered aspects of a variety of molecular genetic pathways that are involved in cell signaling and central to human cancer. The Nobel Committee, in its citation of our work, describes some of these discoveries using the phrase organ development. And by organ development, the committee was referring to the development of the major female sexual organ of the worm, the vulva. And it turned out that the development of the vulva is induced by a signal from the nearby gonad. One cell in the gonad signals other cells to commence vulval development. And what we did was we analyzed the molecular genetic basis of this signaling pathway, and we discovered that the molecular genetic pathway is essentially the same as a key signaling pathway, the RAS pathway in human cancer. And let me explain that briefly a little bit more. The first human cancer gene was the RAS gene, identified in 1981 by Ed Skolnick, who later became president of the Merck Research Labs. After Ed's discovery, biomedical researchers wanted to define the whole pathway of RAS action. What activates RAS, and what does RAS activate in turn? And the answer to this fundamental question emerged from three superficially, apparently very, very distinct lines of inquiry. Some people were actually studying mammalian RAS using cells grown in petri dishes. Second, there were people who were studying something that seemed totally unrelated, the development of the eye of the fruit fly Drosophila melanogaster. And third was our studies of the development of the worm vulva. In a striking and really breathtakingly rapid fashion, these three completely distinct efforts revealed a common molecular genetic pathway, now known as the RAS pathway, activation of which can cause cancerous growth. And the RAS pathway is central in many human cancers and has probably been the best studied signaling pathway in all of cancer biology. Now, importantly, components of the RAS pathway have become major targets for anti-cancer therapeutics. For example, the EGF receptor activates RAS. It's upstream of RAS in the RAS pathway. And the EGF receptor is the target for kinase inhibitors, drugs like Iressa, Tarceva, Gilotrif, and it's also the target of certain monoclonal antibodies like Vectibix and Herbitox, and many others, in fact. Furthermore, understanding the RAS pathway has helped explain why certain cancers are resistant to these drugs, as well as how drug resistance can arise in the first place. For example, if a cancer activates a gene downstream of the EGF receptor, like RAS, or a RAS effector like RAF, then that cancer is no longer going to be sensitive to inhibitors of the EGF receptor. So in short, and I know this was a long response, our studies of worm development have helped lead to a fundamental understanding of how the RAS pathway works and how anti-cancer drugs work. And importantly, and I think really a major message here is this understanding came in large part by absolutely basic science studies of the development of worms and flies.
Well, that was a fantastic explanation, and you've given us such a clear understanding of this aspect of RAS pathways and how they really play such an important role in signaling cancer. Now, coming to that in terms of fundamental research, because you kept emphasizing the fact that fundamental research has thrown up so much important information on which we built a lot of our cancer understanding, on which, many therapies were developed, on which many molecules were discovered. So, you've often said that basic research is the driver of scientific intellect that can lead to great application and impact. And I'd really like you to elucidate how fundamental research is absolutely essential for academic research to generate and sustain applied research. Because very often, Bob, in our country, we often have a debate and discussion about what is more important. Is it important to invest more in basic? Is it more important to invest in applied? And there's always this conundrum of, if you're a developing country, is applied research more important, or do we need to invest in basic research, and so on and so forth? So someone who's actually come from that background of fundamental research and who's applied fundamental research to many aspects and big impact making investments in applied research, I'd really like to hear your words and share your counsel on how should we go about investing in research.
Yeah, well, I think the 30,000-foot answer I would give to that question is like any investment portfolio, it should be diversified. And in that context, there should be some basic and some applied. But the question then is how much in each, which, of course, again, in any investment portfolio can be a complicated question. I think that the studies that we've done of this microscopic worm are really illustrative in the context of the power of fundamental basic research. Our breakthrough discoveries weren't made with the goal of trying to cure or even understand a disease. Our work was absolutely fundamental basic research. And when I began, neither the generality nor the application of our efforts was at all clear. We didn't study people or even another mammal. We didn't target any disease. And I had no way to know if what we found would be relevant to any organism other than a tiny worm. Nonetheless, our studies established mechanisms that are universal amongst animals, and our findings have helped provided the basis for new treatments for a broad variety of human diseases, including, as we've discussed, cancer. And I think the basic message here is simple. Fundamental research, discovery-based research, can lead not only to interesting findings and be intellectually rewarding, and I think those are important human goals in and of themselves, but they also can lead to insights central to disease that would be very difficult to reach by targeting that disease for study. So I really do think basic research is the major driver of scientific knowledge. But the question you ask has a second component, and that is government officials, for example, have to make decisions about how much should go to basic research and how much should go to applied research. And, oftentimes the decision is we want deliverables, we want metrics, and that's where people go to. And I think from what I've said so far, it should be clear that there's a cost to that. The cost is in making really discoveries that are unanticipated. That can't be done in such a directed way. But then the argument comes, and I'm sure you've heard it yourself many times, is leave the basic research to others. You know, the US, very wealthy country, lots of basic research. Let the US do the basic research. And here, wherever here happens to be, we will take those findings and apply them and make our own investments basically applied. I think there are two fallacies with this reasoning. First of all, by doing that, there would be no finger on the pulse. Even that applied research program would have to lag behind because of the lack of contact with new technologies and new discoveries. It would just make things much slower and basically leave all that other progress to others. Secondly, by focusing research entirely in an applied way, there wouldn't be individuals who have been trained understanding how breakthrough discoveries come about. It wouldn't happen in the same way. You wouldn't have the workforce that would be capable of basically doing the things that are needed even to move that applied research. So I think the answer, as I said right at the beginning, comes to a combination. And there has to be some significant amount of fundamental research coupled with some significant amount of research focused to the interest, the particular interest of let's say a country like India.
Oh, thanks, Bob, because this is always a conundrum that a country like India faces. I believe that governments must invest in basic research because a lot of the applied research has to be also done by industry and entrepreneurs. And that's something which we need to inculcate. And I wanted to now shift gears, therefore,
Can
to
I just
entrepreneurs.
can I respond to that for a moment?
Sure,
Because
sure.
the notion of leaving research to companies is another topic that often comes up. And the answer, as you know as well as anybody, is companies aren't going to do research if they can't see where it's going to go. Such research may invariably lead to major breakthroughs, but if those breakthroughs aren't within the context of the company's business plan, they won't do the company any different and any good. So the basic research has to be societally driven. It can't be company driven because it's not in the company's interest to do that.
No, absolutely. I mean, I think we're saying the same thing. What I mean to say is that,
Yep,
you've got to use, government spending in investing in basic research because no one else is going to invest in that research.
absolutely. I'm
And
agreeing completely.
yeah, and I think what I'm saying is that once that research becomes visible for application, then industries are ready to basically grab that opportunity and translate into application. That's why I said that it's very difficult to get any industry or any company to invest in basic research, whereas it makes a lot of sense for academic research to focus on basic research. And most of these academic institutes obviously seek government funding. So I think,
Mhm.
and that's where I think government also has to understand what you said, that you need to spur that scientific intellect through basic research and understand science at a much deeper level to be able to do justice to application. I mean, that's really the fundamental
Mhm.
principles of science. So, I mean, we can keep discussing
Yep.
this, but thanks for your, sharing your thoughts on this subject, which is going to be extremely valuable for people listening to this fireside chat to pick up on. But since you've actually spoken about this analogy of portfolio allocation and saying, okay, you've got to, if you've got a pot of money, then you've got to allocate a certain amount to basic research, a certain amount to applied research. So let's now shift gears to entrepreneurship because, you've been a very, very unique scientist, if I may say so, because you not only did great science, but you translated science to entrepreneurial efforts. Bangalore is often called a startup city. I'm sure every researcher in the audience would love to know how they can leap from their labs into an entrepreneurial ecosystem. And Bob, you've founded and joined many companies in the biotech space. I'm sure everyone would love to hear about that.
Yeah, well, I can't really tell anyone how to leap from academia to industry, but I'm happy to define and describe my own path, and perhaps there's some lessons in that. I have to say, my trajectory from worms to biotech came as much of a surprise to me as my path from IBM and computers to biology. It was 1986. It was 8 years after I became an assistant professor at MIT that I was invited to join the scientific advisory boards of two biotech startups. And I had these invitations, and soon after I got these invitations, I was walking from MIT to Mass General Hospital. I've been involved with Mass General for many years. And I was walking with my good friend, and I call him a wormy colleague, Jonathan Hodgkin, who later became the professor of genetics at Oxford. And I told Jonathan of the two biotech invitations I had, and I also told them that, of course, I'd be turning both down because I was a pure scientist. And Jonathan, who is as pure an academic as anyone I've ever known, stopped in his tracks. We were halfway across the Longfellow Bridge, and he looked at me and he said, "Don't you ever want to do anything useful?" And that took me aback. And I started thinking about it. And then I decided, okay, I'd give it a go. And I said yes to the Cambridge company. And I did that even though the plans for the California company looked better. But I knew I could walk to the Cambridge company, and the California company was going to be more of a pain. So I became involved in biotech. I have to say, my entrepreneurial start would have been far more lucrative if I had chosen California, but so be it. I had dived into the world of biotech. I should add that the next time I had a Massachusetts California choice, I did choose California, not because it was sunnier, but because it again offered a greater chance of success. And this time, as a result, I did better financially, and I got a lot of frequent flyer miles. What I found was that my first biotech experience was exceptionally interesting and stimulating. I learned a lot. I learned to think in different ways about different topics, and I became hooked. I love the idea of trying to see how to apply discoveries in basic biology to making a difference to patients. And it's really that difference to patients that's at the core of what any good biotech or pharmaceutical company is doing. And I have to say, I've never looked back. I've now been involved with 14 biotech companies, of which I co-founded four. For 14 years, I was a member of the scientific advisory board of Novartis. For the past 16 years, I've chaired the medical and scientific advisory board of a major US venture capital company, MPM Bioimpact Capital. And I've had really the enormous privilege of having played a significant role in the development of multiple US approved drugs. I think I've also helped drive some substantial market caps. And in the process, again, I've learned an enormous amount. I've met some fantastic people, and I've had fun. I've just had an enormous amount of fun. And I guess if there's any message I would convey from all of this, it's keep an open mind and be opportunistic because you never know what some new adventure might offer.
Thanks, Bob. It's also been a pleasure to have you as a fellow board member on PureTech, where I think we had a lot of fun too. You know, so that's been a great opportunity for us to learn about your various entrepreneurial, associations. I mean, the fact that you've co-founded entrepreneurial companies is also something that people ought to listen to. In fact, as you know, startups are often, companies that need to have mentors. And of course, you've had many scientific mentors and people who've inspired you, as you say, along the way. And perhaps you'd like to start with the late Obaid Siddiqui, which who I know is someone you knew and is a link to Bangalore.
So let me put Obaid in a bit of in a context of my mentors, because as you say, I've really had the enormous good fortune of having had fantastic mentors. I mean, Bob Solow in economics, who shared an office with Paul Samuelson, another fairly well-known economist. Jim Watson and Wally Gilbert were my PhD advisors. Sidney Brenner and John Sulston were the two people I worked most closely with as a postdoctoral researcher. All five of those received Nobel Prizes. But in addition, there are many others who have provided me with counsel and have become friends. And Obaid Siddiqui is amongst this latter group. Sage, mentor, and friend. Obaid was a pioneer in the field of molecular biology. His work helped define the genetic code. And then later, he was a pioneer in the field of neurogenetics. He worked with Seymour Benzer, another major figure in molecular genetics. And together, they did some of the first electrophysiological studies of mutants of the fruit fly Drosophila. Obaid was from the generation before me, and he was very close friends and collaborated with one of my close MIT mentors, Maury Fox. Maury taught me genetics when I was a senior at MIT. He was my professor. And then many years later, I actually taught that same genetics course, co-taught it with Maury. So full circle. Now, Obaid often visited Maury in Cambridge, and I got to know Obaid over lunches during those visits. And I think we really hit it off and became friends. And as a consequence of those discussions, Obaid invited me to visit him at the Tata Institute while he was still in what was then Mumbai. And that was 1985. And I really enjoyed that visit. I met lots of people. I learned lots of things. I had some great food. I have to say, I squeezed the visit there between a meeting in Rome and teaching a course in Kyoto. So it was the only time I went from Boston to Boston, going all the way around. Um, Obaid always struck me with his calm demeanor and very reasoned approach. I didn't see anything was ever going to really perturb him. He just knew how to handle situations. And I've often consciously tried to mimic Obaid's style. He was better at it than I am. And I have to say, despite the many miles between us and the admittedly infrequent nature of our interactions, I always considered Obaid a mentor and a friend, and I miss him.
Thanks. That's very touching, and I'm sure people who know Ubaid, and he was very well loved and respected in Bangalore. So I'm sure they really appreciate what you said. So finally, let me now turn to technology. The Bangalore Tech Summit is about technology convergence. So let me ask you an obvious question. Computational power in the 1970s, when you did most of your most important research, was fairly rudimentary. If you did have access to high-end data analytics, AI, and other, advanced computing, how differently would you have gone about your experimental work? And I guess, you know, in conclusion, what is your advice to biologists on how they ought to think about technology convergence? I know these are pretty sort of